The aim of our Living with Diabetes podcasts is to share information and inspire, and we endeavour to ensure that the information provided in them is accurate. It's not a substitute for medical or health advice from a healthcare professional who's aware of the facts and circumstances of your individual situation. We always recommend that advice be sought from your GP or diabetes nurse before making any changes to medication or before using any products or services referenced by DRWF. DRWF will not be held liable or responsible for failure to seek competent medical or health advice from a professional who's familiar with your situation. Views, information or opinions contained in podcasts do not necessarily represent opinions, standards or policies of DRWF. The inclusion of details about products or services does not indicate DRWF endorsement of them.
Hello and welcome to the latest Living with Diabetes special podcast looking at research supported by Diabetes Research and Wellness Foundation. I'm Claire Levy, and in this edition we're looking at an interesting research project which measures the effects of kisspeptin on insulin secretion and the growth and survival of beta cells. I recently spoke with Dr. James Bowe to discover more.
So I'm Dr. James Bowe. I'm a reader in endocrinology and diabetes in the Department of Diabetes at King's College London.
So you received a non-clinical fellowship back in 2008. At the time, what did this award mean to you and your career?
So at that time, I was a postdoctoral researcher. So I finished my PhD in 2006. And I had done my first postdoc moving into diabetes and doing this sort of research. And the fellowship, it sort of meant a couple of things, I guess. First of all, it kind of gave me the opportunity to actually take ownership of a project and actually be able to take the initiative and independently sort of plan the experiments. And I think that was sort of really useful in terms of laying the groundwork for then sort of subsequently going on to sort of build an academic career and to sort of establish my own lab. Being able to sort of have that sort of stepping stone in terms of being able to take ownership of a project while at the same time having the support of sort of supervisor and colleagues around me. And then also in the research sense, in terms of the actual kind of research I was doing, that research that I did as part of my fellowship sort of laid the groundwork as well for the sort of research that I've then gone on to do in terms of my sort of subsequent career, having got a lectureship and having started to sort of build up my own research group. The work that I did within that DRWF fellowship really sort of set me down that path and has kind of set the tone for the work that I've done ever since, really. So
how has that funding helped your research career in the next few years, or do you think we've probably covered that?
I suppose getting the fellowship, so as I sort of hinted at before, I guess, getting the fellowship really was the basis for me then subsequently at the end of the fellowship. I applied for a lectureship position at King's, and I think me being successful in that and me being able to sort of successfully apply for a lecturing position was in sort of large part maybe sort of based on me having sort of successfully got that fellowship and through that experience. But then also the work that I did and the results that I got through that fellowship that I guess we'll sort of talk about a bit more in a minute, provided the basis for then sort of subsequent grant applications to then sort of take on my own PhD students, my own postdocs, and start to build my own independent lab.
Is there anything in particular about the DRWF funding that's different to other funders?
I don't know if it's a planned approach or a plan different. I mean, personally, I've always found the DRWF very approachable and also very supportive of early career researchers. So I think they were sort of very supportive at the time that I got my fellowship, but also subsequently after my fellowship and once I'd started to establish my own research group, I also got a couple of bits of sort of smaller funding from the DRWF, some non-clinical funding and also a sort of pump priming funding, which was really useful in terms of sort of starting up new projects and starting up new bits of work. And so, yeah, like I say, in my experience, I think they were really supportive during those kind of early stages of my career as I was sort of trying to transition from being a postdoctoral researcher within a lab to establishing my own research group and getting my own research projects up and running.
So can you tell us the title of your research now?
The title of my 2008 DRWF fellowship was defining the physiological role of kisspeptin in islet function.
Can you tell us more about kisspeptin?
So kisspeptin's it's an interesting hormone. It's an interesting signal. It's best known for its role in reproductive function. So it, as a sort of side thing, it got its interesting name. It was originally sort of discovered in a town called Hershey, Pennsylvania in the US, which is, for anyone that doesn't know, it's sort of the American equivalent of Cadbury's, where they make sort of Hershey's chocolate. And one of their most famous exports is these little chocolate drops called Hershey's Kisses. So when researchers in this town discovered this new signal, they named it after the town's most famous export and called it kisspeptin. So that's where it got its slightly odd name. But it was discovered in the early 2000s. It was found to have a really important role in puberty and in controlling adult reproductive function. And individuals with mutations or defects in kisspeptin or the kisspeptin receptor effectively don't go through puberty and have no adult reproductive function. So the vast majority of the work looking at kisspeptin over the past 20 years or so has really focused on this very important role in terms of regulating reproductive function. The reason that we became interested was because there's also very, very high levels of the kisspeptin receptor within the islets in the pancreas. And at the time when I sort of started off down this kind of research path, there really wasn't any research looking at why that might be. And it wasn't obvious at all why the islets might have these very high levels of the kisspeptin receptor. So in my kind of early postdoctoral research, before I started the fellowship, I was sort of looking a little bit about what kisspeptin might actually do to the islets. And I'd shown that it sort of seems to stimulate insulin release. But the fellowship was then kind of really aiming to build on that and to look at that in a bit more detail.
So where is it found in the body?
So kisspeptin itself is found in various different regions. It's found in the brain, and most of the effects on reproductive function and puberty and those kind of things are effects within the brain. But it's also found in sort of various different tissues, and the receptor is found in various different tissues. Like I say, the receptor and also kisspeptin itself are found in the islets within the pancreas, so that's why we were interested. But there's also very, very high levels of kisspeptin that are made by the placenta during pregnancy, and lower levels in various other tissues. I think there's sort of lowish sort of levels in the liver as well, in the lymph nodes. I think also the testes and the ovaries. And a few other tissues. But like I say, most of the research has focused on that role in reproductive function. But it is expressed in various other different tissues, and so there have been, and increasingly, now that we understand the effects in terms of reproductive function a little bit better, there's increasing interest in trying to work out its involvement in other different systems. And so obviously I'm particularly interested in the islets and effects relating to diabetes. but there are other groups out there sort of interested in the role in other different tissues as well.
And is it fully understood?
I wouldn't have said it's fully understood. So the role in terms of reproductive function is pretty well established and pretty well understood. There are some details to exactly how it works, and some of the more detailed kind of mechanisms of how it works are still a little bit unclear. But in terms of reproductive function, I think it's pretty well understood what the main roles of it are. In terms of other tissues and in terms of other systems in the body and potential involvements there, I think that's probably much less understood. But I would like to think that, I guess, over the course of the research that we've been doing and over the research that sort of some other groups have been doing, that slowly we're sort of starting to understand the role of kisspeptin in the islets and in diabetes a bit better.
So what have you demonstrated with your research about pancreatic kisspeptin?
So in terms of pancreatic kisspeptin, so as I sort of mentioned briefly before, in my early postdoc career, before I got the fellowship, I'd already tried treating islets and treating the pancreas with kisspeptin, and we'd shown that it stimulates insulin release and has various other different effects on the islet function in the pancreas. But the big question that I really wanted to look at for the fellowship was the question of why it does this. Just because kisspeptin sort of seems to stimulate the islets, seems to stimulate insulin release, what the purpose of that was in the body, and what the kind of, I guess, the sort of what biological states kisspeptin might actually sort of play a role in, and why a signal that is most associated with reproductive function might have this additional role in terms of stimulating insulin release. And so what we started to do, and the path that we went down, was really looking at whether kisspeptin might play a role during pregnancy. And part of the reason for that was because under most normal circumstances, most of the time, the levels of kisspeptin circulating in the blood are extremely low, virtually undetectable. But the one exception to that is that I sort of mentioned briefly before that kisspeptin is expressed in very high levels in the placenta during pregnancy. And it turns out that during pregnancy, levels of placenta increase by thousands of fold in the maternal circulation. So we thought that because pregnancy was one of the very few sort of states where the islets would actually be exposed to very high levels of kisspeptin, then maybe kisspeptin might play a bit of a role there. And to take a sort of step back, I guess, in terms of why that might be interesting. So during pregnancy, what you normally have during a normal, perfectly healthy pregnancy is you normally have a gradual increase in insulin resistance across pregnancy. So there's almost a sort of a mild pre-diabetic state during pregnancy. And that's completely healthy. And the reason why that's useful is because it means that the mother takes longer to absorb glucose from the blood. And what that does is it prioritizes the glucose supply to the fetus. So it means that the growing fetus can take up more glucose from the blood. So it's quite a beneficial mechanism. But obviously you don't want that insulin resistance to go too far, and you don't want it to develop into diabetes. So in parallel with that increase in insulin resistance, the islets in the pancreas also have to adapt, and they have to proliferate, they grow in size, and they release more insulin during pregnancy. So I guess the question that we were interested in is sort of what are the signals that tell the islets to adapt? How do the islets know that they're in a pregnant woman and that they need to grow and they need to be making more insulin in order to compensate and to help cope with the demands of pregnancy? And so our sort of theory, I guess, was that maybe kisspeptin is one of the signals that's involved in communicating from the placenta to the islets and communicating that the islets are in a pregnant woman and that they need to adapt. They need to increase their insulin secretion. They need to grow to meet that increased demand. So that was really what we were aiming to look at. We sort of did that in different ways, and in fact, that sort of turned out to be the case. So if you take normal non-pregnant mice, I should say I'm not a clinician. I'm a sort of basic scientist. So all of my research was really sort of non-clinical, based around animal models. But if you take mice and you administer a level of kisspeptin that you would normally see during pregnancy, then what you see is their islets increase their insulin secretion, they grow in size. You see a lot of the effects in response to kisspeptin that you would normally expect to see during pregnancy. And similarly, if you look at it the other way around, if you take pregnant mice and you administer a kisspeptin antagonist, so if you take pregnant mice and you block the effects of the normal kisspeptin that would be present in those mice, then you reduce the adaptation to pregnancy. So if you block the effects of kisspeptin, then pregnant mice, their islets don't release as much insulin, they don't grow to the same extent during pregnancy, and they become glucose intolerant. So that was really the kind of the main findings that came out of the fellowship, this kind of establishing this idea that kisspeptin might play this important role in terms of communicating from the placenta to the islets, to communicating about the sort of pregnant environment and communicating to the islets that they need to adapt in order to cope with the demands of pregnancy. And if you lack that signal, if there's that signal is blocked in any way, then that can potentially cause issues in terms of the mother's ability to cope with those metabolic demands.
Mm. So how are the hypothalamus, placenta and endocrine pancreas involved in that long-term regulation of energy balance? Is really in that sort of interaction between the placenta and the islets. The hypothalamus is certainly involved, and as I sort of mentioned briefly before, the hypothalamus sitting at the base of the brain is where kisspeptin is kind of involved in reproductive function. And the hypothalamus is also very much involved in sort of glucose sensing and various other mechanisms that talk to the islets. But my main interest has pretty much been in that communication between the placenta and the islets during pregnancy. That communication, I guess, is obviously most important during pregnancy itself. So once pregnancy comes to an end, obviously the placenta is gone. So I guess there isn't a sort of long-term communication between the placenta and the islets beyond pregnancy. But it's sort of worth noting, I guess, that that pregnancy and impaired glucose homeostasis during pregnancy particularly does seem to have quite long-term effects. So just because the placenta's gone, the consequences of that sort of placenta islet communication, the placenta pancreas communication, the consequences of that can be sort of longer reaching. So for example, it's reasonably sort of known that sort of in women who develop gestational diabetes are much more at risk of developing type 2 diabetes later in life. And so that's something that more recently I've become interested in, the sort of longer term consequences of impaired glucose tolerance during pregnancy on later life risk of developing type 2 diabetes. There's a kind of question there, I guess, about whether the gestational diabetes is causative for the later life type 2 diabetes. Does the fact that an individual has developed gestational diabetes actually cause
the later life development of type 2 diabetes, or is it just that individual's pancreas and that individual's islets have a greater susceptibility to developing type 2 diabetes, and maybe they would have developed type 2 diabetes even if they'd never been pregnant? So those are the kind of questions that we've sort of become more interested in looking at more recently.
And what did you propose about the beta cell kisspeptin and how it was involved in regulating islet function at times of changes in metabolic demand, such as starvation, obesity, pregnancy, and type 2 diabetes?
So as I say, my sort of earlier work, and most of my work has been focusing on those different things, mostly on pregnancy. And so I guess our hypothesis that we've sort of been looking at and have subsequently found is that kisspeptin release from the placenta sort of increases insulin synthesis in the islets, increases the amount of insulin that the islets release in response to glucose. Kisspeptin seems to stimulate the proliferation and the growth of the islets during pregnancy. And all of these are sort of mechanisms that really mean that when the islets are placed under increased demand, when the insulin resistance in the body requires sort of higher levels of insulin to be released in order to prevent the development of diabetes, that the islets are able to cope with that and they're able to meet that increased demand. So that's kind of been our main focus. We have kind of started looking at sort of other states such as obesity, type 2 diabetes, those kind of things. Our focus has been on pregnancy because, like I say, the placenta releases so much kisspeptin that during pregnancy specifically, it's quite unique that it's a state in which the islets are exposed to huge amounts of kisspeptin, and it seems to play quite a major role. But we have kind of more recently been looking a bit at states like obesity and type 2 diabetes, and there's other groups out there that have looked a bit at the role of kisspeptin in obesity and type 2 diabetes as well. And there does seem to be maybe a less important role for kisspeptin in obesity and type 2 diabetes, but it does seem to have effects there as well. So that's certainly something that we're interested in going forward.
Yeah, your project measured the effects of kisspeptin on insulin secretion and on the growth and survival of beta cells. Did this information gained from the studies identify new ways of modifying islet function with potential therapeutic applications?
So it did. So I sort of mentioned very briefly earlier that after my fellowship, I then also had some sort of other funding from DRWF as well. And one of those bits of funding that I also had from the DRWF was basically a kind of a non-clinical funding to do a sort of small translational study. So this was in 2013. I got some funding from the DRWF for a project that was called Plasma Kisspeptin in Pregnancy and Gestational Diabetes. And so this was a small translational pilot study, basically looking in pregnant women. This was in collaboration with colleagues at King's College Hospital. And so this was taking samples from pregnant women who were undergoing routine glucose tolerance tests as part of pregnancy in order to try and diagnose gestational diabetes. And so we were interested in looking at whether there was any difference in kisspeptin levels between women who developed gestational diabetes and those who didn't. And so what we essentially found was that, firstly, that women who developed gestational diabetes or who were diagnosed with gestational diabetes had significantly lower circulating levels of kisspeptin than women with healthy pregnancies. The other thing that we found was that women with lower circulating kisspeptin levels during pregnancy released less insulin in response to a glucose challenge. So as part of the glucose tolerance test, these women were given oral glucose, which obviously causes an increase in your the circulating insulin levels. But in women with low circulating kisspeptin levels, there was less of an increase in insulin release in response to glucose. And women with high kisspeptin levels had more of an increase in insulin release. So this sort of indicates that, obviously, as I say, I'm mainly a basic scientist, so most of my research is done in animal models, but we obviously, mouse pregnancy is not the same as human pregnancy. And so we wanted to kind of see if this mechanism that we had identified was actually relevant in human pregnancy as well. And so really this seems to indicate that kisspeptin also plays a role in human pregnancy, and maybe that women with low circulating kisspeptin, that may be one of the reasons for the development of gestational diabetes. I certainly wouldn't suggest that kisspeptin, low circulating kisspeptin, is the only reason why women develop gestational diabetes. There's lots of different factors, but it may contribute to the development of gestational diabetes. So going on from there, this isn't something in terms of sort of clinical therapeutic applications. That's not something I personally have taken forward, because not being a clinician, that's sort of not my speciality. But I'm aware of kind of clinical colleagues who've taken this forward. And it might have sort of clinical or therapeutic applications in a couple of ways. So partly one of the issues with gestational diabetes, is that it can only be diagnosed once it's developed. So gestational diabetes is generally diagnosed by glucose tolerance test once the gestational diabetes has developed. There's a lot of interest clinically in sort of screening studies for whether you could screen earlier in pregnancy to identify women that were particularly at risk of developing gestational diabetes, so you could intervene earlier. Because in theory, the earlier you can intervene, the better the outcomes. And so I know of clinical colleagues who are sort of currently looking into developing sort of screening strategies, testing different sort of markers that you could measure during pregnancy to try and identify women at risk of gestational diabetes. And kisspeptin is one of the markers they're interested in looking at. It will probably turn out, it's very unlikely that there will just be a single marker that you can measure that will identify risk. But maybe by looking at a screen of, say, 20 different markers, you could look at that sort of screen of different markers and identify women who are particularly at risk of gestational diabetes. The other approach in terms of sort of therapeutic applications would be, if we say sort of maybe some women who develop gestational diabetes, one of the issues is low levels of kisspeptin. So could we just give those women kisspeptin in order to treat the gestational diabetes? That's a difficult one, because there's obviously very understandable clinical concerns about giving sort of novel therapies to pregnant women because of the potential risk of side effects on the development of the fetus. So the sort of ethical process of developing new therapies for use in pregnant women is a difficult one. But that said, I'm aware that there are currently sort of clinical trials going on to test whether kisspeptin might be therapeutically useful in diabetes more generally, and in type 2 diabetes. So even though we think that kisspeptin actually in the body is most involved during pregnancy and involved in the islet adaptation to pregnancy. In principle, the way that it works, by stimulating insulin release from the islets, by stimulating the growth of the islets, in principle, all of those things are effects that would be extremely useful in type 2 diabetes as well. So even though our focus in terms of the actual role of kisspeptin in the body has mainly focused on pregnancy, there's a lot of potential there that it might be therapeutically useful in type 2 diabetes. And so that's something that is ongoing, and there are actually sort of studies in humans to test the clinical therapeutic potential of kisspeptin as a anti-diabetic therapy ongoing at the moment. I guess often with sort of basic science research, we obviously do a lot of work in the lab trying to understand mechanisms and looking in animal models or cell lines or tissue samples. And so it's really nice to be able to sort of see the early stages of that actually translating into human studies, because as nice as it is to treat diabetes in mice, obviously what we really want to do is to try and understand the mechanisms in humans and to develop sort of research that's going to be useful in terms of treating diabetes in humans. And so it's sort of been really nice to actually sort of be able to see the early stages of that translation from the animal models. And seeing that sort of actually does translate into human studies as well. And to think about how that sort of initial DRWF fellowship has actually really provided the basis for a lot of the work that I've done since. Both provided the basis for me getting my lectureship and in a career sense, but also the basis for the kind of research that I've been doing ever since, really. And since doing that project and looking into that role of kisspeptin in terms of regulating islet function during pregnancy, since then that's really been the basis for my current work, is sort of expanding outwards from that in terms of looking at other different aspects of pregnancy and how the islets adapt to pregnancy in response to different signals and other projects. And all of that really you can kind of trace back to that sort of initial DRWF fellowship, which kind of sent me down that path of looking at gestational diabetes and how the islets adapt to pregnancy. So if people are thinking about
donating, what would you say to them about why it's so important to support research?
I think it's really important because the basic science research in terms of trying to understand some of these mechanisms and trying to unpick how the islets work using animal models or using cell samples or those kinds of things. I think it's sometimes maybe difficult to sort of see how some of that kind of quite detailed mechanistic research is going to sort of extrapolate in years to come into actually sort of clinically or therapeutically sort of useful work. But I think in sort of one of the nice things in my experience is sort of seeing that translation and seeing how what started as trying to understand how a signal was involved in regulating the islets and trying to understand the mechanisms of how that signal was involved in sort of regulating islet function in the body, how that has developed in terms of identifying this potential role in how the islets function during pregnancy, identifying that this is a mechanism that's not only relevant in mice, but also relevant in humans. And now seeing, even though I'm not so involved in the clinical side of things, now sort of seeing clinical colleagues taking that forwards and looking at potential therapeutic uses of kisspeptin, looking at the possibility of kisspeptin being used in biomarker screens for early identification and diagnosis of gestational diabetes. So that kind of sort of funding of basic science research, looking at those mechanisms, really does. It sometimes takes a few years, I guess. It's a sort of gradual process, but it really does sort of translate into kind of future clinical benefits and future sort of real world benefits that hopefully will improve diabetes care. And sometimes it's sort of difficult to predict exactly what those benefits are, but I think funding basic science research is really important, because it's the first step down that pathway of making sort of clinical sort of leaps forward in terms of developing new treatments and therapies.
And I suppose without charities that wouldn't happen.
Not really, and certainly not to anything like the same extent. And I mean, you could really trace that back. I wouldn't claim to be the only person that's kind of like looked at the role of kisspeptin in islet function and looked at those things. There are other groups that have been looking at it, but yeah, I mean, the work that sort of, I guess, started going back with that DRWF fellowship that I did, looking at linking kisspeptin and pregnancy. I guess that work, looking back, was sort of the starting point for the current kind of like clinical studies that are being done now, in part. And that work wouldn't have happened without that charity funding. And I'm sure that's not by far from the only case where that's true, that's going to be mirrored across lots of different projects. There'll be people who are doing their sort of non-clinical fellowships right now, whose work in a few years' time down the line may well develop into different sort of clinical projects or different projects that might have therapeutic applications. And so, yeah, I think without those early steps, it sometimes is a bit difficult to kind of see the link and to sort of see how these basic science studies in cell lines and animal models are going to translate into therapy. But without that first step of trying to kind of understand those different mechanisms and trying to understand how these signals work, without that first step, and often those sort of studies are funded by charities, then you're not going to get the information feeding through that then a lot of the clinical studies are based on. So I think it's sort of critical, and I think, as you say, sort of charity funding plays a huge part in that.
My thanks to Dr. James Bowe for talking to us about his work.
Diabetes Research and Wellness Foundation. Staying well until a cure is found.
Look out for more episodes of our special podcast discussing research supported by DRWF and the advances being made. To discover how you can continue to help the charity, together with the latest fundraising events, news and information, it's all on our website at drwf.org.uk. I'm Claire Levy from Diabetes Research and Wellness Foundation. Until next time, thanks for joining us on Living with Diabetes.
Living with Diabetes is a Blue Aurora media production for DRWF. Copyright 2023. Diabetes Research and Wellness Foundation. All rights reserved.